Let me introduce Professor Jim Groombridge. So Jim is a mainstay of DICE, as it's lovely here seeing faces from the whole history of DICE, from the people who've been here for a week to people who've been here much longer. Jim has really been a real foundational person within DICE for all those years. He's a fantastic colleague, and he also does fantastic research in really cool places. So it's an absolute pleasure to invite him to give his talk today so that we can hear a lot more about his research. Thank you very much, Jim.
Thanks very much, Bob. Do I have to switch the presentation?
Oh, yes, sorry.
That's all right.
I always like to give a good introduction and then run away and leave it.
I just need to find it for a second. Here it is.
Sorry, it's just
It's just hidden, isn't it?
This will be edited out for YouTube.
Here it is.
There we go.
I found it. Fantastic. Okay, well, thank you very much, Bob, for the opportunity, and it's lovely to see you all here. Well done for staying the course. I know this is the second hefty talk in as many hours, so I'm very grateful to you to be here. And it's nice to tell you a little bit about some of the work that we've been doing on some bird populations, and this work extends back many, many years. For those of you that don't know my background, I'm a species conservation biologist, so I work with endangered species, but I also run our genetics laboratory, the Conservation Genetics Lab here at DICE, and so we've been doing some genetic and more recently some genomic work on some of the bird populations I'm going to talk about today. So I want to sort of mix these two ideas together in terms of looking at endangered species, but also what can we learn when we apply genetic methods to those free-living wild populations. So this is about a story, if you like, about a reintroduction project which started back in 2006 and involved a reintroduction, we'll go from there. So first of all, I should just include some acknowledgements and some thanks. So the genomic work that we've done in the last couple of years involves a whole team of collaborators from the University of Copenhagen. So Dr. Hernan Morales from the University of Copenhagen and Georgette Femerling from the same university, and Kok van Oosterhout, who's a professor in evolutionary genomics at UEA, and Rachel Bristol, who is a sort of a world-renowned expert in avian reintroductions, and she lives in the Seychelles, but she's been involved in almost all of the iconic bird reintroduction programs in the world, and she was heavily involved in this project as well. But as well as the genomics, we also need to thank the field teams that have made the reintroductions that I'll talk about possible, and these have been funded by a number of Darwin Initiative projects, so UK government funding, with a whole suite of different project partners from RSPB. Importantly, we've got the government of the Seychelles here, but also Nature Seychelles and other key conservation NGOs on the island, as well as other players, the RSPB and Durrell as well. So whole teams of people made a lot of this practical reintroduction work possible. So to switch now to think about the underlying concept behind this, the sort of the scientific framework, if you like, we need to appreciate that most endangered species undergo some form of population bottleneck, and that's the word we use to describe a situation where a population becomes rare and the number of individuals goes down. And the reason why we talk about it as a population bottleneck is because not all of the genetic lineages that might have existed at the beginning when the population was of a healthy size, not all of the genetic lineages make it through the population bottleneck. If we're lucky enough to conserve the species through reintroduction efforts and captive breeding and so on, then the population recovers. But of course, the alternative outcome is extinction through time. And from a genetic point of view, the same terminology, a population bottleneck, has relevance as well, because what we find if we simulate population bottlenecks for a particular species and a particular demographic profile of how rare that population becomes, we find that the genetic makeup, all of these coloured marbles represent different genetic alleles or different genetic variants in a population, essentially genetic variation, which is key to the viability and the persistence of any species. We find that the genetic makeup of populations becomes very different when a population has been exposed to a very severe population bottleneck. And there's lots of, the whole theory of small population biology has basically developed in the last sort of 30 or 35 years. And more broadly, we know that all species of conservation concern, particularly those on the red list, the IUCN red list, they will have endured some level of population bottleneck. It might be a long, elongated bottleneck, or it might be a very short, sharp bottleneck where the population has dramatically declined in very few generations. But nonetheless, they all have something in common in that there is a loss of genetic variation as that population passes through a bottleneck. So I want to focus for this talk on this species, the Seychelles paradise flycatcher. This is a species that is endemic to the Seychelles, and it was one of the Seychelles' last critically endangered species. The Seychelles, as a country, has an amazing track record in saving its endangered bird populations and species, and there are 5 or 6 different species which it has successfully restored, and one of the last species to become sort of non-endangered as a consequence of successful conservation effort was the Seychelles paradise flycatcher. This is a female, and they build their nests on the ends of these, the fronds at the ends of the tree branches on the takamaka trees in the Seychelles. This is what the males look like. They have these amazing streamer-like tails, and if you are lucky enough to go to the Seychelles and see these birds, they are remarkably easy to see, and they're some of the first birds you will notice because the males are flying through the forest with these streamer-like tails, and they are quite amazing. As a critically, what was relatively recently a critically endangered species, they are remarkably accessible. If you work with Arabian leopards, you don't see a great deal of those, but you can pretty much see, 4/5 of the world population of flycatchers in the better part of a few days. Very accessible species, very fortunately. So where is the Seychelles? So they're a group of islands to the north of Madagascar, quite a long way to the north of Madagascar. And they're a very widespread group of islands, and there are lots of far-flung coral atolls which belong to the Seychelles, but the main core of the island looks like this, the main core of the Seychelles Islands. And this is the main island of Mahé, with the capital city Victoria. And the Seychelles paradise flycatcher, we think, historically would have been on all of the islands in the Seychelles. But if we look in museums, we find that it's present. We see that the museum specimens maybe 100 years ago have been collected from some of these islands here. But we think that it was found across all the islands. But when we began first working with the species, it was only found on Ladigue. So this is a story of localized extinction, where this bird species has become locally extinct from some of the smaller islands, and that the range of the species has contracted over the years. This is a postcard from 1965 of Seychelles Airport. So if you went to the Seychelles at that point, you could have bought a postcard with this amazing, beautiful airport. And it's just to illustrate the changes that this group of islands has undergone over the last few decades. And when this photograph was taken for this postcard in 1965, there were, we estimate, 28 individual flycatchers. So thinking about the population bottleneck, this entire species has become locally extinct from some of the outer-lying islands, and at one point on the island of Ladigue, which is the source island for this species, it went down to just less than 30 individuals. So a really narrow, severe population bottleneck. And if we cast around in the literature to look back in time and try and estimate the population sizes historically, we get some of this data. It doesn't look, there's not a lot of information, but we can infer the population's decline might look something like this, going back into the 1800s and before that. But what's interesting is this. This is the upsurge or the increase in the population in the 1990s and early 2000s. And it's quite remarkable to have a species which has recovered seemingly with not a great deal of conservation effort. There was, before we started to do reintroductions of this species, it was notable that these estimates on the island were going up. And this is in the context of an island system where there's lots of development, lots of new airport being built, lots of tourism. Tourism in the Seychelles is the number one economy in the Seychelles. So this is a real interest. Why has a population gone through such a severe population bottleneck? We know that it's likely to have lost a lot of genetic variation, which should compromise its viability. But actually, this population seems to have recovered almost unaided until the early 2000s. So this still a critically endangered species, but nonetheless, if we look in the literature, these are some of the population estimates that are written for this species. You can see the second column here is the year, and the first column is the name of the island where those surveys were carried out. And what we see really for this species is a story of localized extinction from a whole range of different islands until they were only found on La Digue in 1965. And then surveys on that island subsequently recorded this increase in the population size. So again, this is really unusual. There are very few cases where populations have become so rare and yet have recovered with very little or almost no effort by captive breeding and reintroduction and so on. So in the year the 2000s, this was a critically endangered species, endemic to the Seychelles, restricted to just a single island. And La Digue is an island that you can ride around on a bicycle in less than a day. There are almost no cars. There's a human population of about 2,000 people. There's a single school. It's a tiny island, and yet the world's population existed there. There's none in captivity anywhere in the world. The population decline, we think, is because of habitat loss and introduced invasive species, rats, cats, the usual culprits that conservationists know very well. And so the government of Mauritius put together an action plan to recover this species by reintroducing birds to different islands in the Seychelles where there was suitable habitat. So that was the plan at around about 2004. And I joined DICE in 2003 and began to become interested in species like this, which have been through population bottlenecks, and to see where we can help restore those populations. So this is a aerial view of La Digue Island. Again, very small, beautiful. This is where the 5-star hotels and the beautiful white beaches are that you see on the postcards in the Seychelles Islands. It's an amazing place to go. One of the things we needed to do to start with before working with this species was to explore a little bit about how special, how evolutionarily distinct this species is from all of the other paradise flycatcher species that are found on the other islands in the Seychelles, in the Indian Ocean. And this slide shows a picture of some museum specimens of the different species of flycatchers that are found within the Indian Ocean. There's the species in Madagascar. In fact, there are 2 different morphs of the Madagascan paradise flycatcher, and that's those ones here. And I've laid them out broadly geographically against that map. So this is the Réunion flycatcher, the Mascarene flycatcher that's found on Mauritius, the Comores flycatchers. Comores Islands have their own taxa of flycatchers, and then the Seychelles flycatcher. And what really stands out is this remarkable tail of the males. The males are all on the right-hand side there. So, and it's really quite remarkable. I had to put this picture in on the left. This is Rachel Bristol holding a male with an extraordinary tail. I mean, these things, you know, the evolutionary processes that have produced these long tails in the male is remarkable. So Rachel and I began working on this project to understand how special, how evolutionarily distinct the flycatcher is in the Seychelles, and we collected DNA samples from a variety of museum specimens and also blood samples from different species of flycatchers, and sequenced some of their DNA and produced a molecular phylogeny, which is a way of trying to infer the levels of evolutionarily distinctiveness between different taxa. And what you see at the top there is the Seychelles flycatcher, which has the long branch length here illustrates that it has been isolated on the Seychelles for a very, very, very long time. And it's one of the most evolutionarily distinct taxa within the group of flycatchers within the Indian Ocean Islands. Anyway, against that backdrop, we decided the plan with the government of the Seychelles was to start to establish reintroduced populations on different islands. And at the time, Dennis Island in the north was considered the most appropriate island to put flycatchers on it. So a lot of these different islands, they have slightly different habitats. Some of them have rats on the islands, which impact on small bird populations. These flycatchers are insectivorous, so they need to have enough food supply as well. So we did a lot of habitat surveys to figure out which islands were the most suitable, and Dennis Island ticked all the boxes. It was also a rat-free island because it is managed by a Seychelles-run company as a privately run island where they've eradicated all the rats, and it's now a haven for introduced or reintroduced populations of birds. And so the decision was made that we could translocate some of the birds from the only place where they were in 2007 to Dennis Island. So that was the plan. And that's what Dennis Island looks like. It's about a 25-minute helicopter ride from La Digue. And the decision was made to take about 20 birds from La Digue to Dennis Island. So about 10% of the world population we were going to catch and put somewhere else. Anyway, just a few nice photographs to set the scene. So we did the translocation in November 2008. This was a Darwin Initiative funded project, and we're there with our mist netting team all on bicycles because that's the only mode of transport, and we would pedal off to try and find these flycatchers, which are sort of zooming around in the forest around us. The guy in the middle, Andrew Greenwood, is from Wildlife Vets International, which is the organisation we worked from a veterinary capacity for guiding us through the reintroduction. And so we were mist netting. We caught the birds using mist nets in the forest, and we involved all of the organisations, but also the local communities on La Digue. So the Diguois are very passionate about their flycatchers. They were only found on La Digue, and therefore they were felt to be really important. As a kind of an element of national pride or regional pride on that island, and they were really great in helping the teams to do the translocation. And this is Rachel helping do some of the mist nets. They've got a female there caught in one of the mist nets. They're just taking it out. So we caught up 23 birds, so about 10% of the world's population, and we put rings on them. We took a blood sample, importantly, for genetic work, and then the plan was to translocate them over to Dennis Island. And here we are. Some elements of the project were really, there are lots of technical specifications, and other elements we had to go right back to basics. And this was one of those things we had to go right back to basics. So here we are. We've got some cardboard boxes from the local supermarket. We're making some Seychelles Paradise Flycatcher translocation boxes, and to house the birds, we held them overnight, and then we flew them out the next day. Holding these birds is not something you want to do for very long, because they are, small passerines tend to be quite flighty and quite stressy. So we wanted to be as hands-off as we possibly could. But there we are, getting ready to creating places to keep them. And so we're off to Dennis Island. That was our plan. And so we got ready to move the birds. We get quite a logistical operation to get everyone in the right place, catch the birds, people to move them to the local football field on La Digue, which was the helicopter landing zone, and then we get them in the helicopter, fly them over to Dennis Island. And we're all ready to do this. You can see Rachel there with a colleague of hers helping her. And we're about to put them in the helicopter, and we're all ready for that. And then you can see the slight look of desperation on my face. This was captured when I'd just been told the news that I hope, Jim, it won't be a problem, but, and you sort of hear that phrase and you kind of hold your breath. And it came over the radio. We had a team of people on Dennis Island who were going to receive the flycatchers from us. They came through on the radio saying, well, it doesn't happen every year, but this year's been unusual. Every year we get maybe 1 or 2 red-footed falcons that kind of get lost on their ways, migrating from Europe down to southern Africa. We get 1 or 2 every year. Anyway, last night, about 40 of them turned up. I've never had this before, and they're all quite hungry. Anyway, off you go. We look forward to seeing you with your flycatchers. So I'm there sort of trying to mentally calculate what I was going to say to the Darwin Initiative when we just had 10% of our flycatchers eaten by red-footed falcons. Anyway, it's one of those occasions where you think, well, you just have to roll with the punches, basically, and that's a phrase which was coined coincidentally by one of the founders of DICE, Mike Walkie. He said, you just have to roll with the punches in conservation, and that's precisely what I thought went through my mind at that moment. Anyway, here's one of the red-footed falcons. We got there with our consignment of 23 flycatchers, and fortunately, these birds had been on such a long journey. They'd been blown off course, found themselves in the Seychelles, and they were exhausted. Although they were very hungry, they were exhausted. And they were too exhausted to basically fly after birds of prey or passerine birds to feed on. They actually generally fed on the dragonflies and things like that. So we were enormously relieved, but nonetheless, letting go of these flycatchers, having to stand there and go, yeah, it's all going to be fine, don't worry, it's all going to be fine. There was a lot of breath holding going on and sort of crossed fingers behind my back. Anyway, we gave them a last feed of sugar water to give them a bit of energy, and then we released them onto the island. And what happened then was quite extraordinary. They began to breed much more quickly than we thought. They began to set up territories on the island much more quickly than we thought, build nests and lay eggs. And we had our first flycatcher chick fledged from a nest on Dennis Island in March 2009. So that's about 2 years or a year and a half after we'd translocated them there. And we went on to document a few milestones. So we moved 23 birds. We had our first breeding behaviour literally a few months later. So looking at signs of courtship, setting up territories, the first chick in 2009, 5 chicks later that year, and then we had some second generation offspring being produced by 2010. So it was an amazing sort of success from the point of view of looking at the population grow. And the population grew. One of the things that really allowed it to increase in population size quickly was a parallel project where they were removing the populations of introduced minor birds from Dennis Island, because the minor birds were predating on the flycatcher chicks. We were seeing lots of breeding going on, but not a great deal of production at the other end, and it was because they were being predated on. So we had a parallel project that removed the minor birds from the island, and then we saw the flycatcher population begin to take off. And so by 2013, this is a map of the island showing all the different territories. And by 2022, we had a population there of near 180 birds on the island. So really, really strong population, and way beyond the kinds of carrying capacity that we thought the island had when we initially released those 23 birds. Anyway, we began to think about the vitality, the viability of this population, and how luckily we've been that this population has increased after being reintroduced. And so we began to ask some genetic questions about that, thinking back to the bottleneck idea and how, well, this species should be genetically quite impoverished. So how come it's doing quite well, or doing very well, with very little genetic variation, having made it through the bottleneck? Because it went down to 28 birds. So we began to look in museum specimens. These are specimens collected over 100 years ago by naturalists that visited the islands at that time, and they did their ornithology with a shotgun, thankfully, because it's what we have today, are some very rich museum collections. And so Rachel and I set about sampling these birds for DNA, so that we could look at the levels of genetic variation in the DNA from the museum samples and compare that to blood samples from the modern population on the Seychelles today. So Rachel went to the Seychelles, she's lived there for many, many years. She has a family there now, and is a Seychelles citizen. So she blood sampled a large proportion of the source population on La Digue, so we could look at the levels of genetic variation and then compare that to the museum samples. But before we did that, we thought, well, we actually want to know how much genetic diversity that existed on La Digue, how much of that have we actually moved to Dennis Island contained within those 23 birds. And we did some simulations, and this just shows on the bottom axis the number of individuals that you could theoretically translocate, and up the side is the number of alleles, or the number of genetic alleles, the amount of genetic variation that could in theory be moved given the genetic variation in the source population. And so we compared these simulations, which show that, yeah, if you translocate more birds, you'll move more genetic variation. But there's a kind of law of diminishing returns at the top. And so we compared it to the actual 23 birds that we genotyped. We blood sampled them and looked at the variation. And we actually did slightly better than average in our luck that we had for the 23 birds that we chose. But nonetheless, they have been through a bottleneck. They're a genetically impoverished species. So we then began looking at the museum specimens in more detail, and we could piece together the profile, if you like, the temporal profile of levels of genetic variation through time, because each of the museum samples has a label on it with a date, so we can date when those birds were in the population, when they were alive, and we showed that there's been a substantial loss of variation through time as the population became rare, and we're also able to compare the rate of loss historically with the amounts of genetic variation that were lost purely by picking individuals and plonking them on a different island. You leave some genetic variation behind. So thinking about the bottleneck idea, we know that not all the genetic lineages got through the bottleneck, that this is a genetically impoverished species that we're working with. And so fast forward now to about 3 years ago, by which time the technology for looking at genetic variation in genomes has vastly transformed and become much more detailed in what we can do with it. And this on the left here, this is a DNA sequencer which was about the size of a sort of, you could probably fit it on this table just about, and that would sequence DNA for you, about 1,000 bases of DNA sequence per run. And today, the same piece of equipment is about the size of is the size of this. It's about the size of a thumb drive, and you can load your DNA sample in the top, plug it into your laptop, and it will sequence the DNA for you, and many thousands of times faster and in greater depth. So the technology has moved on, and that has meant that the cost of sequencing DNA plummeted, the blue lines on the left, through time, and as a consequence, sequencing whole genomes has now become sort of revolutionised the conservation discipline in terms of conservation genomics. And that's the arena really where we're now operating to see how we can use genomics to inform conservation on the ground. And so the last slides want to show you some of the most recent results we have from that. So alongside that, this sort of torrent of genomic data which has exploded in the last sort of half a decade, we're now, it's now possible to sequence, all species on Earth is the eventual aim. But there's a B10K project, which is where they're sequencing the genomes of 10,000 species of birds, and it's now ballooned out beyond that to a single representative of every species on Earth being sequenced. So the advances are huge in that regard. So we began to use that technology on the DNA samples that we had from the museum samples from 100 years ago in the Seychelles flycatcher, but also on the modern day blood samples. These three colours, this is a map of the Seychelles Islands, and the green triangles are samples we collected from the modern samples, these sort of the source samples, and these red and purple represent the historical museum samples that were collected from those two islands before those particular island populations went locally extinct. And we began to analyse these samples, and this graph shows the levels of genetic differentiation between those different populations. And all it shows is that the two populations on Praslin Island and Ladig Island historically are genetically very different from the modern population today. And if we look at levels of genetic variation, one of the popular metrics is heterozygosity, which is a measure of genetic variation. And the graph on the left shows, again, the same colours. These are the historical museum samples. This is the level of individual heterozygosity. Each dot is a museum sample. And here, in comparison, are all of the blood samples of the modern Paradise flycatchers today. And you can see this remarkable drop in levels of genetic variation. We're dealing today with a genetically very impoverished population. And in more detail, this graph illustrates across the genome of these birds. It shows the average levels of nucleotide diversity for the modern birds, which are right down here, low levels of diversity compared to museum samples. So we then sort of delved a little bit deeper, because one of the things you can do with whole genome sequence data is to backcast, to estimate the size of the population back in time. And that's really, really valuable, because that allows us to sort of verify how severe the population bottleneck was in the sort of last few centuries. And the reason we can do that is because we can look at what are called runs of homozygosity, which are basically chunks of DNA, fragments of DNA in an individual's genome, which are purely homozygous. In other words, there's no genetic variation contained within that particular fragment of the genome. And within an individual bird's genome, we can measure the length of those fragments or runs of homozygosity. And it's the variation in the different lengths of runs of homozygosity that we can use to infer or backcast to estimate the population profile in history that would have been required to produce this frequency histogram of the differences in runs of homozygosity that we see in the modern population. So from sequencing whole genomes, we get vast amounts of information. And so we were able to use the information in the top graph that you've already seen to estimate and backcast estimate the effective population size of the Paradise flycatcher just using the modern contemporary genomes. And what that shows is that it estimates that the population went through a very severe bottleneck, but actually it confirms that the presence of the bottleneck in the 1960s or 1970s, and that's validated by the field records. So we know we have a signature of this very severe population bottleneck from the genomes that's validated by the field records as well. So a very severe population crash, and it compares reasonably well with the field estimates that were collected from field reports. And I feel that's remarkable to have a signature from a set of genomes that actually reflects the field data that's collected over the last sort of 100 years or so. Anyway, we started to go a bit further on that, and using the same sort of techniques, you can backcast much further in time than just 200 years. You can backcast maybe 500,000 years or further to see, well, what was the population size like many hundreds of thousands of years ago. And for the Seychelles paradise flycatcher, we know it's been sat on an island, a tiny island, for a long time. And one of the surprises was, from the genomic data, it seemed to suggest that the effective population size has actually been quite small for about the last 10,000 years. But then after that, in time, it was a much larger ancestral population size. And that got us scratching our heads thinking, well, how on earth can you have a population that's been on an island and yet ancestrally has been at such high population sizes? And it wasn't until we began to look at the sort of biogeography of the Indian Ocean and we realised that there were submerged marine platforms which are underwater today, but with sea level changes. In the past, those sea level changes would have been lower, and they would potentially have exposed large areas of land back in time, which may well explain those genomic signatures that we saw. We couldn't fit enough flycatchers onto the Seychelles Islands in what the genomes were telling us until we began to look at the biogeography of the region. And so to get back to, well, how has this population recovered? It's recovered without any real conservation effort until we started doing reintroductions, but it's recovered without any real effort. So we start to look within the genomes, and we can quantify the number of deleterious mutations. These are mutations that accumulate in small inbred populations of species. And using the genomes, we can detect those and quantify them. And here I've got two graphs which show for the museum samples, the museum samples and the modern samples, it shows highly deleterious mutations. So these are really bad mutations that really impact on, we think, the fitness of birds, the ability to breed and produce eggs and fledglings and so on. And what we call LOF, or loss of fitness. These are mutations that do bad things and will impact on the fitness of that individual bird. And these are mildly deleterious mutations. So these are mutations which are actually much more of a problem because they accumulate in small populations, and natural selection is less able to get rid of those mildly deleterious mutations as they accumulate in rare populations. So those are all of the deleterious mutations that we could find in the genome, but not all is equal in the world of deleterious mutations, and it depends on what form they're at in the genome. So if deleterious mutations are found in the heterozygous form, in other words, individuals that have inherited from their parents just one copy of a deleterious mutation, and the other copy is a wild-type allele, then the deleterious effects of that mutation won't be expressed. So most deleterious mutations are recessive. In other words, they're not expressed in the heterozygous form. So the effects of that deleteriousness or badness is hidden or masked from natural selection. But what happens when populations become small is that inbreeding starts to occur, and that allows an environment for those deleterious mutations to become expressed in their homozygous form, which is where individuals inherit two copies of the same allele. And if they inherit two copies of a deleterious allele, then the deleterious effects on fitness, the negative effects on fitness, are expressed. So when we look at the graph of the homozygous deleterious mutations, whereas for all the mutations, we see that the really bad mutations, there's no difference between modern and historical populations. When we look at just the homozygous mutations, we see that there is a very small but statistically significant reduction in the number of really bad mutations in the Seychelles flycatcher population. So natural selection has been effective to some extent, and it's been able to purge some of that variation out of the population, even though natural selection is not really operating very well because it's a small size population. However, the real problem is this, and in particular these guys. So these are mildly deleterious mutations. They have a bit of an impact, a negative impact on fitness, but not so much that natural selection is able to get rid of them to select them out of the population, and therefore they accumulate in the surviving population that comes out of a bottleneck. So essentially there's these really bad mutations have been purged from the population as a consequence of purging, but genetic drift, which is the random change in the frequency of alleles, particularly when populations become very small, and they they drift to a higher frequency, and that's what causes inbreeding depression. The depression in fitness, number of eggs laid, number of fledglings hatching, and so on. So the purging of highly deleterious mutations. And so then we went on to estimate, well, this is almost the last two slides, you'll be happy to know. So the top graph, we were able to put aside all the genomic data and to actually simulate, using computer simulations, simulate the flycatcher population through time since the 1800s. And the red line there, time's on the bottom axis at the graph at the top, and the red line is our sort of simulated population size. And we then varied we simulated the same population bottleneck, but for a flycatcher population which has 5 times historical population size and 10 times historical population size. And we wanted to see, well, what's the impact of having a different population history on the ability to survive in terms of deleterious mutations. And so we use a software, I say we, Georgette and Hernan Morales, who are the real wizards behind SLIM analysis, was simulating performing these computer simulations. And we can partition what's called the genetic load, which is the load of deleterious mutations that which cause all these inbreeding depression problems. And what we found is that if you're a species and you have a history of small population size back in time, a bit like the Seychelles flycatcher over the last 10,000 years, then you have a much lower genetic load and also a lower masked load, which is the proportion of variation which is hidden from natural selection. In comparison to if you're a population which has been at a historical population that's much larger. So what we're seeing is that these simulations are telling us that actually looking back in time is actually really important to inform us about the probability of how viable a population is in the future. But if we look at another component of the genetic load, which is called the realised load, and the realised load is where the masked load becomes converted to realised load by inbreeding. So essentially the realised load is the deleterious mutations that are open to being expressed through by natural selection. And what we see here is a very different story. So through time, from the ancestral population size, when the population collapses, so the bottleneck starts, if you've been sat on an island for a long time, you get away with a much lower realised load than if you have had a much higher population size. And even more so when the recovery phase of your history starts. And then natural selection, as the population increases in size, natural selection kicks back in, and we see an overall reduction in the realised load. But what we're beginning to learn is that the differences in the ancestral population size has profound impacts, we think, on how we can project populations, whether they're going to be viable in the future or not, is often, we think, determined by their history. And this is all through simulations, and then using genomic data to validate those models. So this is where we think the flycatcher is, that it's benefited from this long period of isolation which has purged all of these deleterious mutations and allowed it, we think, to recover relatively unaided on the Ladigue Island. So where does this all leave us? This is my almost last slide. So today the flycatcher is listed as vulnerable, population of about 400 birds, but we know that this species is genetically very, very impoverished, and we've done additional work that I'm not including here, where we've looked at the future viability, the future fitness of the population into the future. But I think what it illustrates is that with genomic data, we're now able to learn a huge amount about, not just about the current population, but inferring about the population sizes in the past and how that really helps inform us how we move forward with these populations in the future. Okay, and that's my last slide. Thank you very much.